All right, in the last segment, we talked about the basic structure of the nucleobases, the nucleosides, and their nucleotide analogs in both the deoxyribonucleotides and the regular ribonucleotides. I want to show you now a little bit about oligonucleotide synthesis, or how this happens in biology. We're just focusing in on the chemistry, not necessarily on the templating and synthesis of complementary strands. And please understand that whatever we do here is going to necessarily be an oversimplification of what you will later learn in biology and biochemistry. So essentially, to make an oligonucleotide, we need a linkage between the 3' hydroxyl group of of the one nucleotide, and then the 5' phosphate of the other nucleotide. So we're going to want to make a bond between the 3' oxygen on this AMP and the 5' alpha phosphate of the GTP. And so the AMP is going to be our nucleophile, the GTP is going to be our electrophile. To understand the chemistry here, you need to get used to the idea of this as which is a pyrophosphate group, as a good leaving group. Sorry. And so likely in the synthesis, a base removes a proton from the 3' hydroxyl group, so it's nucleophilic. Then that base attacks the alpha phosphate group, the alpha phosphorus atom. I'll draw that arrow here. Lone pair electrons attack the phosphorus. The mechanism of this reaction, there's evidence that it proceeds via an addition intermediate elimination mechanism. That is, you get a intermediate in which the 3' hydroxyl oxygen is bound to the alpha phosphorus before the leaving group leaves. Zooming in just on the relevant portions of the molecule, that intermediate would look like this. I'm going to use an R here for, well, when drawing stuff in biochemistry, it's never clear how much of the molecule you should draw to get your point across. I'm already regretting how much of the molecule I'm drawing here. I'm going to abbreviate the rest of it like this. Then over here, we're going to have the next ribose ring and our G base. And then over here, we're going to have the pyrophosphate leaving group. And I'll point out that a lot of times magnesium is coordinated to that pyrophosphate leaving group to make it an even better leaving group. If you're following along, though, I've missed another atom to phosphorus. So this intermediate is sort of a pentavalent phosphorus intermediate. Then electrons kick down and the leaving group leaves. And that creates a new bond between the A nucleotide and the G nucleotide, and that bond is mediated via this phosphodiester linkage. So we have an AG dinucleotide, and this bond is a phosphoester, this bond is a phosphoester. Together, this functional group is called, probably need to move it to the side. Together, that segment is called a phosphodiester. And that is how individual nucleotides are linked together in oligonucleotides like DNA or RNA. A couple of things you need to be aware of. Notice that the oligonucleotide, like a lot of other polymers in biology, has directionality. There is a 5' end of the oligonucleotide that has a 5' phosphate group. And then on the other side of this dinucleotide, we have a 3' hydroxyl group. So DNA has directionality. DNA and RNA has directionality. It's got a 5' end and a 3' end. DNA synthesis, based on what I've shown you, always takes place in the 5' to 3' direction, because it's the 3' hydroxyl that's acting as a nucleophile. You should be aware that in oligonucleotides, people will talk about the sugar phosphate backbone. That's the ribose, the phosphodiester, and the next ribose, and so on. And then the bases, which are hanging off the ribose ring via these N-glycosidic linkages. Now, I've got a figure from your text which shows an example of polynucleotide where you can see this in greater detail. Instead of just being a dinucleotide, this is a tetranucleotide. You can see, like the dinucleotide above, we have a 5' phosphate and then a 3' OH. This is a deoxy oligonucleotide because we lack the 2' hydroxyl, so this is from DNA, not RNA. We see the sugar phosphate backbone labeled in gray, ribose deoxyribose ring, 3' oxygen phosphodiester, 5' oxygen ribose, 3' oxygen phosphodiester, 5' oxygen, and so on. That's the sugar phosphate backbone. And at each of the anomeric carbons via an N-glycosidic linkage, we have one of these nucleobases, cytosine, adenine, thymine, or guanine. And so this poly or oligonucleotide could be written as deoxy CATG. And that would tell you, written left to right, 5' to 3', would tell you the sequence of this oligonucleotide. So let's talk about the sort of structures or conformations that can be adopted by these oligonucleotides. And this was elucidated by Franklin and Watson and Crick in the 50s based on X-ray crystallographic data. Here is an image of a DNA duplex, and you can see that it involves two oligonucleotides wrapping around each other in what's called a double helix. And you have on the exterior of that double helix the so-called sugar phosphate backbone. The carbon atoms of the sugar are colored gray and the hydrogens are colored white. The phosphoruses are colored orange, and they're bigger than carbon because phosphorus is one row down on the periodic table from carbon. And then the oxygen atoms are all red. So you can see alternating phosphate, that's the phosphodiester, ribose, and so on. In the interior is where the nucleobase is. Here is a purine nucleobase, hydrogen bound to a pyrimidine nucleobase across the DNA double helix. If you look at the double helix, you can see some interesting features. there are two grooves. There's two sets of spacings, a much wider spacing here between sugar phosphate backbones, where you can see exposed nucleobases, and then a much narrower spacing here between sugar phosphate backbones. That's typical of the DNA double helix. The large spacing is called the major groove, and the small spacing is called the minor groove. And you can see, though it's not clear, it's hard to see what the individual structures of these purine and pyrimidine bases, that the pairing of the DNA duplex is maintained by complementary hydrogen bonding across the duplex. So A hydrogen bonding with T, C hydrogen bonding with G, and so on. This is the structure adopted by DNA, which lacks the 2' hydroxyl group. The structures adopted by RNA are much more varied. RNA can engage in a double helix. RNA can form hybrid duplex double helices with DNA, but RNA can also adopt some other interesting structures as well. Okay. There are various what are called A form and B form and other forms of duplex DNA which differ slightly in the conformations and the sizes of the major and the minor groove. You may learn about that in biochemistry. We're not going to worry about it in organic chemistry. Another thing you will learn about potentially in biochemistry is the major and minor grooves expose a lot of hydrogen bond donors and acceptors. As we look at the edge of a base pair, you can see there are some, such as this nitrogen and this oxygen, that are involved in hydrogen bonding, and there are others, such as this nitrogen, which do not appear to be involved in hydrogen bonding. And those are accessible for interaction with other things that might like to bind DNA, like histone proteins, which wrap proteins up into tight chromatin structures in your chromosomes, or transcription factors, which can bind to and recruit transcription machinery to begin the process of DNA transcription. All right, so we've talked a little bit about DNA structure. I want you to read briefly about DNA replication, transcription, mRNA translation / protein synthesis, and then PCR. These are all very important subjects in modern molecular biology. What you read is mostly going to be a survey. There's not that many questions. I can ask you about them, and I don't really have anything meaningful to add relative to what the text will tell you. I need to leave that to your future biochem and molecular biology professors to expand on the details there. But I do want to tell you a brief story that's not in your text about how chemists have contributed to oligonucleotide synthesis. So we'll call this, instead of biological oligonucleotide synthesis, we'll call this chemical oligonucleotide synthesis. And this is important because the chemistry is so efficient that if you were to go online to a nucleotide synthesis company and type in the sequence of an oligonucleotide you wanted, they could make it for you and they could purify it and ship it to you by tomorrow. So the chemistry is really efficient, and it really has enabled the revolution in molecular biology and ongoing developments. People need oligonucleotides for almost any project in molecular biology. So I want to show you the chemistry is so well worked out that people don't necessarily worry about it anymore. It wasn't always that way, of course, and I just want to give you a sense for how it's worked out. So chemical oligonucleotide synthesis happens on solid phase. For the same reason that peptide synthesis happens on solid phase. We'll say, though, chemical oligonucleotide synthesis is more efficient than peptide synthesis, and you can make much longer oligonucleotides than you can peptides. So the chemistry is a little bit unusual. We're not going to worry about the mechanism. This represents the solid phase or the solid support, and there is a linkage between the solid support and the oligonucleotide. And we'll draw this with now the 3' end of an oligonucleotide. Let's suppose it's DNA. Now attached via this linker to the solid support. Here will be the base, which we'll call B1. That's the first base in the sequence. The user would decide what it is, and you would need to have put the oligonucleotide on the resin to begin with, on the solid support to begin with. We're not going to worry about how that happens. Then the 5', so the 3' hydroxyl group is linked to the resin. The 5' group is actually protected via a protecting group. I'll go ahead and give you the abbreviation. It's called dimethoxytritol. Its actual structure isn't important for what we want to talk about. You do a deprotection reaction. This is using trichloroacetic acid as a strong acid, which removes the dimethoxytritol protecting group. This is sometimes called detritylation. Again, do not worry, please, that you don't know what the structure of the tritol group is. That particular feature doesn't matter. But what this does is this unmasks the nucleophile. Now, do you remember biological oligonucleotide synthesis? It was the 3' hydroxyl that was the electrophile, and the 5' phosphate, the 3' hydroxyl in oligonucleotide biological synthesis was the nucleophile, and the 5' phosphate was the electrophile. Chemical oligonucleotide synthesis switches that. We now use this as the electrophile. This 5' hydroxyl is the nucleophile. I'm getting ahead of myself. I'm sorry. What's the electrophile? It is something called a phosphoramidite. And the functional groups here are going to be kind of unfamiliar to you. And so we're not actually going to worry about the chemical reaction or the mechanism, what's going on necessarily with the bond formation. The electrophile has a 5' hydroxyl group as well, but it is protected. And then the electrophilic portion of the molecule is here. You have a phosphorus bound to two things. Here, let me illustrate. So the 3', the electrophile is our phosphoramidite. Our phosphoramidite electrophile is the next nucleotide that we want to attach. And we want to make a bond between the nucleophilic 5' hydroxyl group of the resin-bound or solid support-bound nucleophile and the phosphorus. What's here, which I will highlight in gray, is not yet a good leaving group, but we're going to use a set of reagents to activate it, to turn it into a good leaving group. That involves an acid catalyst / nucleophilic catalyst known as a tetrazole. I won't show you that reaction mechanism. You don't need to worry about it, but the process eventually causes this group in gray to leave and to be replaced by the oxygen of the 5' hydroxyl of our nucleophile. So after the activation and the coupling steps, what we have is as follows. We have a new bond between what used to be our nucleophile and the phosphorus. Now, if you're following along at home with what's going on here, we've made the new linkage between the first nucleotide and the next nucleotide. But this doesn't look like a phosphodiester linkage, in part because this phosphorus is reduced relative to phosphate in a phosphodiester. So the next step in the synthesis is called the oxidation step. This involves iodine and water, but it basically oxidizes the phosphorus up to the right oxidation step, putting another oxygen there. And then you can, so we've made a new bond between the 5' hydroxyl of our resin-bound nucleotide and the phosphorus of our next nucleotide. That new bond is sort of illustrated here. And now you can see something that begins to resemble a phosphodiester. This functional group is a protecting group for the negatively charged phosphodiester backbone. And then we are ready for another cycle. We can do another deprotection step. Which will unmask the 5' hydroxyl. Then we'll bring in the next phosphoramidite. Which we'll call B3, and after activation, coupling, and oxidation, we'll get our third nucleotide unit on there. That would look like this. And you can see how we're now prepared for another cycle of deprotection, next monomer, activation, coupling, oxidation. And so this iterative process gradually strings one nucleotide on to the other, but the reaction's pretty fast, pretty efficient, and you're able to make large nucleotides with defined sequences, which has been really, really, really important in molecular biology. I want you to be aware of this. I want you to be aware of the structural features. I'm clearly not expecting you to know what the DMTO protecting group is or what the acid is doing. We could talk about that, but it's not really worth our time right now. I don't expect you to know much at all about phosphoramidites other than to recognize that this is the starting material or monomer for adding the next nucleotide in the sequence. But I want you to have the idea that after activation and coupling, you generate this kind of intermediate, which needs to be oxidized to get you to something that looks like a phosphodiester. At the end of the day, if this was where we were done after we had finished the reaction and we wanted a free 3' hydroxyl, we would do deprotection and cleaving. We would need to deprotect the DMT group, the dimethoxytrityl group. We would need to cleave from the solid support. And then we would also need to cleave the phosphodiester protecting groups. And what that would give us, which we could then purify and isolate, would be this trinucleotide. With now those phosphate backbone protecting groups gone, with the 3' hydroxyl now free from the resin, and with negatively charged sugar phosphate backbone as shown. So this is how you can synthesize oligonucleotides in the lab, and as I said, there are a lot of companies out there that are very, very good at this now. All right, with that, that concludes our discussion on nucleic acid chemistry. And next time we will move on to metabolism.